Hello, I'm Dr. Natalie Castellana, CEO at Abterra Biosciences, and I'm going to share with you today a case study in which we attempt to answer the question, to B cell or not to B cell in antibody discovery. At Abterra Bio, we're a provider of VHH antibody discovery and sequencing services, and we've been analyzing numerous camelid immune responses for more than the past 6 years. From an immunized llama or alpaca, there are several technologies that can be used to identify VHHs. Everyone is likely familiar with phage display, which was the first technology used to isolate heavy chain-only antibodies. You may also be familiar with single-cell screening techniques because they've been widely used in other species. One challenge, of course, for the single-cell approach is the relative infrequency of the heavy chain-only encoding B cells. Llamas produce both conventional heavy light chain antibodies and heavy chain only antibodies, and the proportion of B cells producing the heavy chain only can be anywhere from 10 to 30% of the total population. A third approach for VHH discovery, and one which we've adopted and optimized at Abterra Bio, is a focus on serum antibodies. While comparisons of these different approaches are often done in the general sense, we haven't seen a realistic comparison of traditional phage display to the serum-focused approach to the same antigen from the same starting immunization. For these two approaches, we can use them to mine the exact same immune response. And if we do that, do they recover the same things? So a little bit of background on Alicanto. So why serum? Why not focus on B cells? Well, one big reason is that serum antibodies are abundant and accessible. There's 11 orders of magnitude more antibodies in blood than there are B cells. Even if we account for their difference in size, there's still more than 9 orders of magnitude difference in terms of antibodies in blood than there are B cells. Blood is also antibody dense. So we can screen millions of antibodies per project because in 30 mLs of blood, there's more than 6 million different antibodies that are at a detectable level. And finally, in any immunization-based antibody discovery approach, the serum antibody titer is the go, no-go decision point on whether or not the immunization can be stopped, needs to be extended, which animals to select. So why throw away all of that information and that screened serum? With Alicanto, we retain that the serum and analyze those antibodies directly. So in this particular project, we immunized a llama with SA-Cas9. Throughout the immunization, we collected PBMCs. We worked with a CRO to do phage library construction. From that starting library, before any panning, we used our next generation sequencing technology and analysis platform to give us a baseline repertoire of what that phage library looks like. We also sampled the PBMCs directly. So from the same bleeds that were used to create the phage library, we also directly did NGS analysis. The first thing we compared was the mutation count. So what I'm showing here are histograms of mutations across the different sequences in the repertoire. We compared the phage sequences to the 2 different isotypes that we observe of heavy chain-only antibodies, the short hinge and the long hinge. In the phage process, because you're cloning the variable region into phage, we can't tell whether a particular phage sequence came from an IgG2b or IgG2c isotype. But based on this analysis, it looks like the predominant, the isotype that is best represented in the phage library is the IgG2b. So here on the left, we see that the phage distribution of mutations is quite similar to both the week 9 and the week 13 PBMC sequences that were of the IgG2b isotype. And that is typically the more abundant isotype that we see in PBMCs. Compared to IgG2c, phage is a little bit less mutated. And in general, this is what we see across background repertoires, is that the IgG2c sequences tend to be a little bit more mutated than IgG2b. The second question we asked was, does the phage library use the same germline V genes as the background PBMCs? And what we found was that the phage library is more likely to incorporate conventional V genes than the background repertoire, but not excessively so. So 14% of the repertoire uses conventional antibody V genes versus 10% in the background repertoire. And this likely has more to do with the primer design and how the phage library has to be constructed. So we didn't just sequence the library. Of course, we work with our CRO to do panning. So after 3 rounds of panning, we recovered some hits. So 88 colonies total were selected. And then after sequencing, those collapsed to 25 unique sequences from the phage library. Because sequencing is a big part of what we do at Abterra Bio, we also sequence the library after each round of panning. So we have, in addition to the pre-pan library, we have after round 1, round 2, and then round 3, from which the final colonies were selected. So if we compare across those 4 repertoires, pre-panned, 1 round, 2 round, 3 rounds, you can see that there is a population of mutated sequences that is enriched across the panning rounds. So what I'm showing here is the same histogram of mutations from the pre-panned library in pink and the blue phage library after the 3rd round of panning. And you can see that there is a little hump here of more mutated sequences showing that potentially there is enrichment happening here. We can look at individual clones, not just at a repertoire level, at the repertoire level. And so we can look at enrichment, the enrichment ratio over the pre-panned. And what I've highlighted, each line here represents an individual VHH in the library that was detected in all of the different samples, all the different panning rounds. And then the red ones were highlighted as being ones that were strictly increasing in enrichment across the panning rounds. So we just picked the top 20 VHHs as our NGS enrichment hits, and those collapsed to 13 different CDR3 sequences. So our first question was, how do these NGS enrichment hits compare to the traditional phage hits? And the answer is they didn't look at all the same. So this looks like eyeballs, but it's actually a Venn diagram. So looking at CDR3 sequences in the NGS enrichment set, which was 13 different CDR3s, versus the phage hits where there were 25 different CDR3s. Even if we relax our definition of CDR3s to be 85% similar, there's nothing similar between the NGS enrichment and the phage hits. So that was kind of an interesting outcome for us. And of course, using NGS to augment your traditional antibody discovery technology is something that everybody should be doing. And this is just kind of demonstrating how you can get 50% more candidates if you include NGS sequencing with your phage process. Now, the approach that we use at Abterra Bio is called Alicanto. So we collect the same PBMCs, but from the immunization, we also collect antibodies from serum. From the PBMCs, we run our standard repertoire sequencing process. So we amplify the heavy chain only transcripts. We use next generation sequencing to analyze them. But on the serum side, we select for the heavy chain only antibodies using the traditional protein A, protein G approach. We pull out only antibodies that bind to the antigen. We subject them to mass spectrometry. And then our Alicanto proprietary software integrates the B cell repertoire together with the antibody sequencing data generated in the mass spec to come up with a set of high affinity functional antibodies that are present in serum. So why do we need to use both? So NGS gives us a broad map of everything that is encoded in the B cells. So it's naive things, it's partially matured things. What using the mass spec allows us to do is prioritize. So very few of the antibodies that you detect as B cells are actually being secreted or measurable in serum. And so focusing on highest affinity, not necessarily those that are most transcriptionally active or most expanded, can often be a way to prioritize the antibodies. The second thing that's unique about Alicanto is that unlike the NGS unlike phage display, we sequence before we do selection. And the reason that we do that is because, as you saw in the NGS enrichment and in the phage hits, you come up with, n different colonies or n different positives, but then after sequencing, they collapse down to a much smaller number of different kind of distinct sequences. And that's just how the immune response works. You're going to get lots of sequences that are highly similar to each other, but you don't want to spend a lot of effort testing things that are very similar. So we sequence first with Alicanto so that we make sure that we're sampling the diversity of the immune response. We don't want to oversample big clones that are only single amino acid away. We don't want to miss rare clones that are very distinct. And so with Alicanto, we can verify the diversity that we're delivering and give you a higher chance of success in moving a hit forward. So we ran Alicanto and compared it in the Venn diagram to our NGS hits and our phage hits. We saw good overlap with the NGS enrichment. So 8 CDR3s were found by Alicanto and also by NGS enrichment. But again, no overlap with the phage hits, which was sort of interesting. With Alicanto, because this was, a big blobby protein antigen, we found lots of different CDR3s, so over 300 different CDR3s for this particular project. This is, of course, reporting sequences, and with every Alicanto project, we deliver all the sequences for candidates that we find. But for testing, we ended up down selecting to a smaller set. So we picked 24 of the phage hits, 24 of the Alicanto hits that were disjoint from the NGS enrichment hits. So 13 total CDR3s were tested in the phage hits or in the enrichment hits. We visualize this in terms of expression, binding, and diversity. So these are the 24 phage hits. We reformatted all the VHHs as dimers with an IgG1 Fc attached to them. So many of the phage hits did not reformat in a friendly way. So those kind of could not be considered then for the binding assay, obviously. So 25% of the phages didn't reformat and didn't show expression. And then only 4 out of the 18 that did express had good binding to the target in our ELISA assay. But they were very diverse. So we're not seeing, after we only picked VHHs that were distinct from each other. Looking at the NGS enrichment, all of these expressed really well in the VHH FC format. All of them showed good binding to the target. Of course, the downside to the enrichment analysis was that we didn't have strong CDR3 diversity. So we did see clusters of CDR3 families that behave very similarly. So while NGS enrichment had a high hit rate, it had lower diversity. Then comparing it to the Alicanto candidates. So Alicanto, because we're focusing on antibody proteins that are stable in serum and high affinity, we have a good success rate in terms of expression and reformatting them as the VHH-FC, as well as binding. And of course, we sequence everything before we select candidates, so we know that we're getting good diversity. So Alicanto shows a good balance of diversity as well as success in hits. So just to conclude, what we found with our analysis is that the phage library that we're using has some biases, but it behaves much like a standard PBMC repertoire. We showed that NGS enrichment analysis is a low-cost way to obtain more hits from a phage project, and you should just be doing it by default, as a lot of experts are recommending. And that Alicanto enables a broad interrogation of the serum antibody response, and it can be very complementary to the hits discovered by phage display, including delivering diverse hits that were maybe not captured through that orthogonal method. So with that, I'd like to thank you for your attention, the llamas and alpacas for being so obliging in their immune response, and our whole team at Abterra Bio who worked on this project, and our funding source for this project. Thank you.